enough time for to come. So now we're going to begin the meeting. So thank you to everyone for coming, but an even bigger thank you to Dr. Ujjwal Rathore, we are very lucky to have him here. He's a postdoctoral scientist working at UCSF's Gladstone Institute of Genomic Immunology, where he is working on groundbreaking technology like gene editing and virology. He is working to cure diseases that were previously thought to be uncurable. So thank you for taking time out of your busy schedule to come meet with us and talk with us about your career and the technology in it and how you got there. I myself am very excited to learn how to follow in your footsteps, and I hope the Med 101 members are as well. So this is our agenda for today. Okay, yes, this is Dr. Ujjwal Rathore, who I have just introduced to you. And first, he'll speak a little bit about his career around CRISPR gene editing and viruses.
Hello, everyone. Is it okay if I share a few slides?
Oh, yeah, absolutely.
Okay, just let me share.
I'll give you, I think you have, yeah.
Yeah,
you should
have. Yeah. Okay, so I guess you guys can see my screen. As Himangi introduced me, my name is Ujjwal, and I'm a postdoctoral scientist at UCSF and Gladstone Institutes. So Gladstone Institutes is an institute that is affiliated with the UCSF. And what we do is, our motto is science overcoming disease, which means we want to do science that help us find solution to uncurable diseases, such as cancer, autoimmune disorders, and viral infections like COVID-19. So a little bit, I'll just, give you a little bit of background about myself. I did my PhD at IISc Bangalore in India, and my PhD was in molecular biophysics, which means that I was trying to understand how biological molecules interact with each other and how can we manipulate those molecules. For example, proteins, proteins are the action molecules of the cells. And so how can we, and each protein has a three-dimensional shape, and protein functions in a specific way because of its specific three-dimensional shape. So I was trying to figure out, how proteins attain their three-dimensional shape, and can we change that shape? And can we use this knowledge to design vaccines against viruses? And one of the vaccines that I focused on during my PhD was making a vaccine against HIV. And in 2018, I moved to San Francisco and started working at UCSF. My focus shifted a little bit from vaccines to also looking at finding cure for diseases. So vaccines are usually prevent a disease, right? So if you have a vaccine, for example, for HIV infection, then you are less likely to get HIV infection. But someone who's already infected, and there are like millions and millions of people who are infected with HIV. Right now, every year, close to 500,000 people die because of HIV infection. So you also need a cure for that. And it's been close to 40 years since HIV was discovered. And despite 40 years of research, we do not have a vaccine for HIV and we do not have a cure for HIV. So I moved to Gladstone, to UCSF to figure out new ways to find a cure for HIV. And before coming to that, I'll start with something very basic before I tell you a little bit more about my research. You all know that our body is made up of cells that have specific jobs. And cells contain specific proteins, which are the action molecules that let these cells do their job. For example, neurons, help us process information from outside environment and as well as respond to that, sensory information. And DNA is the molecule that actually encodes these proteins. So sometimes before we get into, gene editing and all these things, I think it's important to understand what a gene is. And the way I like to look at, I'm a foodie, okay? I love food. So the way I look at genes, sometimes when I think about genes, how to explain, suppose you have to go home and explain it to your, younger sibling or someone who is not from a science background. How will you explain genes, proteins, RNA, and all these things and mutations to someone who is not from a science background? I look at a genome. So a genome, or whole, genome is basically a total gene content or total DNA content that we have in cells. So I look at it as a recipe book. So the whole DNA content of one cell is considered as a recipe book, and then each gene is basically a recipe. but the recipe is written in a code that you can't read. For example, let's imagine that it's written in language that you don't understand. Let's say Hindi, which you don't understand. Then although the recipe book is in front of you, you can't read the recipe. So DNA is the recipe, and in order to read it, you have to translate it into a different language that's called RNA. So that's the intermediate molecule. So once you translate it to RNA, then you can read the recipe. So DNA is basically recipe; it's a genetic code, but our ribosomes that make proteins, they can't read that recipe unless it's translated to RNA. Once RNA is there, then that RNA is basically translated to proteins. It's proteins are made out of that RNA code, and ribosomes do that job. And so I would say that proteins, a chef is a protein that does the job and makes a recipe, or you can say recipe. A particular dish is a protein. And this is how I look at these things. Now imagine that in a recipe book, there are you are making a copy of it. Okay, you are writing down a recipe from the recipe book, and you make a few mistakes there. So you know that DNA code is written in 4 letters: A, T, G, and C. Now imagine that instead of writing ATGC, you write AGGC. That's a mutation, right? So when you get a mutation, what happens when the code is changed, and because of which the RNA that is going to be formed from this DNA is going to have a mutation in there, and that's going to form a protein that's not the right protein sequence, and the protein can be misfolded. It might not have its right 3-dimensional shape. So genetic changes or mutations can cause basic changes in protein structure, and that can lead to a disease. So this is what we are trying to explain here. And so there is something very interesting which you people might have noticed and might have wondered at times, that all our cells have same DNA. So for example, if you take a cell from your liver and you take a cell from kidney, they have the same DNA. Your skin cells have the same DNA as any other cell type in the body, but they are so different from each other, right? So they each cell, because each cell, only some genes are expressed in each cell. So let's say, for example, you might imagine that multiple homes in your locality might have same recipe book, but some people are making only one dish from it, following only one recipe from it, and other people are making other recipes from it. They're not making all the dishes. So each cell only, and that's the reason why skin cell is different from an egg or a sperm, right? Because when I was in school, I used to wonder, why can't we take 2 skin cells and fuse them together and make a new human being? If they have all the genes that are needed to make a new human being, if their genes are same between an egg cell and a skin cell, why do you need an egg cell or a sperm cell? Why can't you take 2 skin cells and make them, make a new human being? And the reason for that is because each cell only reads the instruction for the protein it needs, and some genes are expressed and some genes are not expressed in these cells. So as I explained, a mutation in a gene is when a DNA base, a DNA code is changed. So instead of T here, suppose you get A, that's a mutation, and that mutation will lead to a dysfunctional protein. Let me just make it. I just realized that it's not in slideshow. So when that happens, it can prevent a cell from doing its job. A mutation can prevent the cell from doing its job, and that will cause a disease. Now, how could we treat these diseases, right? So it's genetic disorder. There is a mutation in the DNA. How will you treat something like that? To treat something like that, you need tools with which you can change DNA. And a few decades ago, we didn't have any such tools. But now, very recently, we have found a tool using which we can cut any specific location in DNA and paste any other sequence that we would like to do. So using these methods, you can actually treat genetic disorders. And that's what we do. So these tools are called Crispr tools. And some of you might have heard about it, that, and I'm not going to go into a lot of details because you can ask specific questions during the QA session. But so what you can do, you can use this CRISPR tool. So what are these CRISPR tools? Just imagine them to be like molecular scissor. So with scissor, you can cut things, but these are molecular scissors. You can cut DNA with these tools. So you can correct a mutation, and you can prevent formation of a faulty protein, and you can cure a disease. So, but what is basically this CRISPR system? So this CRISPR system comes from bacteria. The structure you see here, that's basically a schematic for a bacteria. And so in bacteria, a few years ago, scientists found that in bacteria, they see these clustered means they were together, regularly interspaced DNA sequences. So you see that this AATT sequence is again repeated. The sequence in green is repeated again and again, right? So scientists found that in bacteria, there are some repeat sequences, and they are getting repeated again and again. But scientists didn't know at that time that what was the function of these these repeats, these DNA repeats. They had no idea what these DNA stretches do. So but after a while they figured out that the sequence between this repeats, like see the DNA sequence between the green region, this multicolored, so here you see it in blue, and so this region, this sequence was always different. And they found out, so when the internet became more accessible, they were able to actually what do is take this sequence that is given in blue here and search it against thousands of sequences, and they found that these sequences which are between this green region, they come from viruses. So bacteria was keeping a region of virus in its own genome for some reason, right? And so they didn't understand what was the reason for that initially, but then later they figured out that what is happening is that viruses, just like they infect us, they also infect bacteria. So there are viruses that infect specifically bacteria. They're called bacteriophages, or phages means bacteria eaters. And and bacteria also has immune system like us. So we have an immune system. When virus comes and infects us, or bacteria comes and infects us, our immune system tries to fight them off. Similarly, bacteria also try to fight these viruses off. So what happens is when a virus comes and infects a bacteria, it injects its DNA into bacteria. And using this Cas9 protein, in bacteria, what bacteria does is it cuts a piece of viral DNA. So what it is doing is it's taking a snippet of viral DNA and stores it in its own genome. So it's like, you see someone who you think is, basically going to cause trouble, right? And so you take a picture of them, you paste it, you paste it in your school. Okay, this person was seen outside the school last time, and there was trouble after that. So be watchful if you see this person and inform the security. So this is what bacteria does. It takes a snippet of, it cuts a piece of viral DNA and store it in its genome. So if it sees this DNA piece again, so suppose it gets infection again, and this piece of DNA comes from a virus, then it just attacks the virus and chops off its DNA. So this was a system which was a part of bacterial immune system. But what we learned from it, so humans are very smart in using things from different organisms. So what we learned is, oh, so this CRISPR-Cas9 system can make cuts in DNA, and it can also insert pieces of DNA at other places. So it means that there are some molecular scissors that it uses to cut DNA and paste other parts. So can we learn about this system and use it to cut DNA where we want? And that's what we did. We learned from bacteria, we learned from viruses how to cut DNA, and develop this CRISPR-Cas9 system, which is, so CRISPR is, I explained, is basically this in genome, this locus is called CRISPR, and Cas9 is a protein that is a nuclease that can cut DNA. So we can use this system to actually cut human DNA as well, to cut plant DNA as well. And you can, use this for genetic editing. So this is what this CRISPR-Cas system is what I use. And this is what I'm showing here, that if a virus, a bacteriophage, which looks like this, comes in, and so the bacteria cuts portion of it and stores those DNA portion into its own genome and use them to recognize virus the next time. So as I was saying, that we can do gene editing with CRISPR. We can use these scissors to cut DNA wherever we want, and we can really change biology using this. And you can imagine like a disease, which is a genetic disorder. Suppose there is a mutation in a gene. That mutation affects a protein that is important for you to see, right? Important for vision. If you know that this is a mutation that is causing blindness, early blindness, then you can actually basically use these molecular scissors to correct that mutation and cure blindness. And that's what was done very recently. Actually, some people were partially cured of their blindness using CRISPR gene editing. So what I do, so what you can do is you can correct a mutation in cells outside the body and then put them back into the body. This is called ex vivo editing. then what you can also do is you can actually edit a person's gene directly in their body. Like I was explaining that blindness was cured in this way, directly injecting this CRISPR-Cas machinery into the eye of a patient and directly correcting genes there. So the right protein is produced and now they can see again. But what we do, so I work in the lab that's called Marson Lab. And what we do is we use CRISPR-Cas9 gene editing to understand the genetic basis of immunity, how our immune system works, how our immune system fight diseases, how sometimes it gets out of control. So we use gene editing to study cancer. We use it to study autoimmune disorders, but I use it primarily to study infectious diseases like viral infections. And so I study viruses which cause disease like HIV, flu, SARS-CoV-2, and any virus that cause a disease. And the way I do this is, so we have around 20,000 genes in human genes. So what I have developed a method using which I can delete all 20,000 genes in human genome. So I can take one cell and delete gene number 1 in it, another cell, delete gene number 2 in it, and then I can take millions of cells and delete all 20,000 genes many times in those cells. And then I take these gene deleted cells and infect those with a virus. Now, viruses are master manipulators. They come with very few genes. For example, HIV has 9 genes in it, and we have 20,000 genes. But if HIV comes into our body, it stays there forever. We cannot get rid of it. Our immune system cannot get rid of it. So, how does a virus do that? A virus, it hijacks our system. It hijacks our genes, our proteins. So what it does is that a virus has 9 genes. HIV virus has 9 genes, suppose, for example. It makes 15 proteins out of those 9 genes. And it uses its own proteins to hijack our proteins. It's like someone comes into your house, okay, an invader, and it hides somewhere in your home. And then it brings its own blanket, and it brings its own slippers and toothbrush. But in the night, the invader gets up and goes down to your kitchen and steals food from your refrigerator. But then to go down, the invader needs supposed to use your elevator or your staircase, and there is a key at your home that he needs to open the door, some doors. So those all the keys he gets from your home. So what we are trying to do is we're trying to figure out what are those human genes that viruses hijack to infect us and to stay hidden in our body. So if, suppose I delete gene number 1 and I find out now HIV virus cannot infect us anymore. Now HIV virus cannot stay hidden. It becomes visible to us. Then we can kill the virus with drugs. So this is how we are using gene editing to actually figure out which all pathways in our body viruses hijack, and which of those pathways we can use drugs to cure the disease. And this is I want to tell you like a little bit about that. I work with a number of these viruses, but mostly with HIV/AIDS virus. And the pandemic started in 1981, and the pandemic is still going on. As I said earlier, that 500,000 people die because of HIV infection every year still. And this is a very old picture. This is from March 2021. So now many more people have died because of COVID-19, but I also work with COVID-19. And I'm happy to answer questions about how we are using genetic engineering specifically to address these things, and your questions about genetic engineering as well. That's it from me, and I'll take questions now.
Okay, thank you. Next, we'll be having a moderated Q&A, which is these are general questions that we've preset, which I believe a lot of people have about your career, because as you know, it's a very interesting career. So what inspired you to follow this career path?
That's a very nice question. So when I was in school, actually when I was in high school, I once asked my biology teacher that the same question, why can't we take 2 skin cells and make a human by fusing 2 skin cells? And my school teacher told me that, I don't know answer to this question, because answer to this question was not known at that time. And he said that, but if you want to find out answer to this question, you have to become a scientist. And that's when I realized that if there are unknown questions, if you have to find answers to those questions, then you have to sometimes become a scientist and find them yourself. But I was also very highly motivated to find solution to difficult problems. So like, for example, treatment to cancer, like solid tumors, like lung cancer, there's no treatment for it. And also, like I used to read about this viruses like HIV and dengue millions of people dying every year, and we have no treatment for these things. So I was motivated by curiosity and a desire to bring a change, and that's what brought me to this path.
Thank you. Our next question is about, I think my screen is frozen. One second. Okay. Our next question is actually focusing on your educational experience. So in general, what was your educational experience like?
So I did my education in India. And after my school, I decided to get into a course that was focused on research, bachelor's in biomedical sciences, where they were basically teaching us a lot about how to do research and like a lot about human body. And after my bachelor's, I got into a program that was an integrated PhD program where I did my master's in biological sciences and PhD in biophysics. So it was an integrated program. And that was, I showed a picture of that place. That place was called Indian Institute of Science. And that place was like exclusively focused on research. So once I got into that institute, I was given an option to choose which department I was interested in doing my, PhD in. And so we were given an option to basically work in 4 different departments. And I did my rotations in biochemistry department, molecular biophysics department, then molecular reproduction and genetics department, and also microbiology. So I went into all these 4 departments and I tried to figure out where my interests lie. And after working in all these departments, what I figured out was that I really like taking challenging problems. And so and there was a lab that was basically trying to understand how proteins fold and take their 3-dimensional shape. So I got in touch with them and I talked with them that can we use some of these things to design vaccines for viruses, because this is something I feel passionate about. And luckily for me, they were already thinking of doing that. So they were interested in that already, and they had money and funding for doing that. So I got involved with flu vaccine design and HIV vaccine design. The way PhD programs, both in the US and in many other countries, the way they work is that you basically do initially a number of courses. You finish a certain number of credits. And once you finish those credits, then you give a candidacy exam. And after that, you become a PhD candidate. And once you become a PhD candidate, then you start doing research on the topic on which the lab is in general working. But sometimes what happens is that you find unexpected things, unexpected results. So you're working on a very specific project, but you discover something that you never thought you'll discover, something new, something really cool. So and depending on which lab you are in, you sometimes get the opportunity to actually pursue that cool result and do other things as well. For example, during my PhD, I was trying to basically, and so this HIV virus, it has a coat of sugar on it, so which it derives from us. So basically, when HIV virus enters into our body, it gets into our self, and it uses our cells as a factory to make more copies of itself. And but it basically, from our genes, from our proteins, it makes a coating of sugar on itself. And because of that sugar coating, our immune system doesn't recognize the virus. Our immune system looks at the sugars and thinks, oh, these are human sugars, I don't need to attack these. So it doesn't attack the virus. Our immune system doesn't attack the virus, and virus survives. So I was trying to figure out how those sugars, what those sugars do, and I was trying to get rid of the sugar from the virus surface. And I found out actually that actually, if you get rid of some of those sugars from the virus becomes more infectious. So basically generated a more infectious HIV virus by trying to learn something else. And that taught us a lot about HIV virus, how to make it less infectious and all. So my educational experience in this field, I think it was very exciting because there were a lot of problems which I was trying to solve, and a lot of problems which my friends were trying to solve, and brainstorming with them for their problems, to help them with their problems and getting help from them. So I think it was a very nice learning experience for me.
That's amazing. Our next question is more focused on your work life. So what do you do in a typical day? Like what activities would you do? What's your schedule like?
Yeah. So right now, it's the way my day works is it's mostly thinking about experiments. So for example, if you, I'll give you a recent example. For example, this new variant of COVID-19 has appeared recently, and it's called Omicron. And this variant is people as it looks more transmissible. It looks like it might spread much faster, and so my typical day goes like thinking about small or big problems and like what problems we want to tackle for that particular project, and then actually designing experiments and doing those experiments. So as a virologist, my day usually starts with wearing a gown, a hazmat suit, and which involves wearing a big gown, 2 layers of gloves, a mask, a face shield, eye protection, a headgear, and then getting into a virus facility where we work with these dangerous infectious viruses. So the facility I work in, it is called a biosafety level 3 facility, which is where you work with really dangerous viruses for which there is either no treatment or they cause very serious disease. So like I work with HIV virus, right? So if you get infected once, so if by mistake when working with the virus, you inject yourself with the virus, like you prick yourself, then you are going to be infected for life, right? So so you have to be very careful. You have to wear that suit. But we have been trained to do that, right? So so basically thinking about an experiment, doing an experiment, analyzing the results, data, then it also involves a lot of meetings. So now I have assistants who help me with my experiments. So a lot of times I don't have to do experiment myself, but I need to discuss with my trainees, my assistants, my technicians, and sit with them and discuss the experiments they are planning to do, to think about how exactly the experiment should be done, what should be the controls, how we are going to interpret the results, how we are going to do the data analysis, and all those things. Also, my day involves writing grants, which is about getting money for your research, because you have to basically give a proposal and say, this is the topic I want to research on, and give it to federal agencies or private donors. And then basically, if they like your idea, they like your pitch, then they fund your research, right? And so that's how basically getting money for our research. And then the other thing also that involves my daily routine is writing our results, and basically writing what we call publications. So when you do research on something, you compile all the results and you publish them so that everyone can read those results, comment on them, and do further experiments and take the field forward. So it also involves writing papers.
All right, your life sounds really busy, but also really fun. So thank you. Which leads me to my next question, which is, what do you love about your job?
Oh, I love a lot of things about my job. So I think the first thing is that I love about is the joy of finding things, right? So whatever you are working on, those are all unsolved problems, new questions. No one has done that before. So the fact that just think about it also give you a little bit of anxiety, but just think about a problem. If you're working on a problem that no one has been able to solve for last 40 years, a cure for HIV, and you're working on it, and then so you have to really bring some new cool ideas. But then the way science works is that you always find something new, right? So you, even if you are not you might not find a cure for HIV today, but you might learn a lot of new things about the virus, and that opens so many new directions of research. So I think it's what I like about my job is it's very unpredictable, it's very exciting. And I think what I also like about it, it brings a change in the society, because you are working on problems that can make a lot of difference. As I was saying, science overcoming disease. So for example, I'll give you an example. When the COVID-19 started, US FDA, they approved certain number of testing kits, those antibody testing kits in the US, and they said that for approving those kits, like testing kits, we are not going to look at your data because we are too busy. But if you tell us that your data is right, like that, those companies which were making these testing kits, if you claim that your data is right, then we are going to just approve your kit. And we found that not to be a good way to move forward, because what if one testing kit works with 60% efficiency, another one works with 90% efficiency, and government and some country buys like few hundred millions of those kits and later find out that they are not working as expected. So what we did was we decided, okay, this is what is needed right now. And we got like COVID patients from UCSF, and we ordered these testing kits from all over the world. And we basically tested those on like confirmed COVID positive patients and negative patients. And we figured out a way, we developed an assay in the lab to test those kits. And we figured out which kits work, which don't work very well. And then we also figured out, suppose if a country has bought a kit that works only with 60% efficiency and another one that works only with 70% efficiency, which kits they can combine to get 90% efficiency. So you test on two kits, you put the nasal swab on two kits and still get good results. So that they don't have to rebuy those kits and all. And so when we did that work and that got published, US FDA had to change their rules. They looked at our data, our experiments, and they said, no, we are not going to approve kits in this way anymore. We are going to actually test and figure out kits the way these guys tested. And that's how we are going to approve these things. So you actually, you feel really good about it, right? That you could make a difference. So I love all these things about science.
That sounds really amazing. Sounds really great. But now, unfortunately, with the good things come the bad things. So what are some downsides of your job?
This is a very important question. So the downsides are, this is not a 9-to-5 job. This is a job which you are passionate about and you do. So sometimes you do it all the time. So and sometimes the boundaries get blurred between rest of your life and the work, because what happens is that you imagine, so right now a pandemic is going on, so it's a very different situation right now. Obviously the scientists will work all the time to develop a vaccine, or because you will not make a vaccine in 3 years because scientists want to work 9-to-5, right? So you will work faster and you would like to bring it to market faster. But just think about normal times. Sometimes what happens is, if you are working on a problem and you think you can make a difference. For example, you can find a treatment for a new type of cancer, for lung cancer. So if you think the stakes are so high, you think, okay, if I make this discovery, maybe we will find a cure for lung cancer earlier, or we'll make a breakthrough that might help other scientists find a cure for cancer. Then it's about just motivated, and you put in extra hours, and you just so I think some of the downsides are, one thing, as I said, this work-life balance is not going to be as great as in some of the other profession where you have fixed working hour from 9-to-5. Here it's like doing your own business where you invest more and then you find out more. So sometimes you just get greedy and you invest more and more. The other downside, I would say, it's a very long career. It takes time to get settled in this career. If you start after your bachelor's, the median time to finish a PhD in the US is 7 years. So 7 years after your bachelor's. If you want to do an MD PhD, that's by definition is around 8 years, which is 2 years of medical school, then 4 years of PhD, and then back to medical school for 2 more years. But most people don't finish it in 8 years. Now that's just PhD or MD. But if you decide to do MD, after your MD PhD residency, then again it's going to take 2-3 years more at least. And if you decide to do a postdoctorate after PhD, then again it's going to take 4 or 5 years, or 3 to 5 years, depending, especially in medicine, because PhD and postdoc in medicine takes time. So it's a very long career. I would say that's why this career is for someone who is exceptionally motivated, is genuinely interested in doing it. You should not just follow someone else, like someone else did it and I also want to do it. It looks cool. You should basically try and figure out if that's for you or not.
Okay, thank you for those insights. One more thing about your career. What is something that you wish you knew before you chose this career pathway?
I think I wish I knew that how important it is to network even in this field, because as a scientist, sometimes you think just that, working on your experimental bench in your lab, that's good enough, because you are reading things and you're doing experiments, you're figuring things out, and so that's good enough. But with time, I realized that just like any other profession, it's very important to network properly in science, because sometimes you are trying to solve a problem and someone else might have a good solution for it. They might not have a problem, but they might have a good solution. They might have a good technology with them that can help you solve this problem. So, but if you're not networking, you don't know people, you're not talking with them, then one thing is you are not going to solve a problem as fast as you can. And the second problem is then sometimes, for such a long career path, you need a lot of motivation for extended periods of time, right? So having a right group of mentors and a group of peers who can support you. And that's one of the most important thing for this career. And I learned that along the way, and I wish I knew it before I started. Yeah.
All right, thank you. This is, I believe, our last question. So do you have any advice for high school students who wish to pursue a career in medicine or just STEM in general?
I think, as I said, that this is a very long career, right? So you need to really understand before getting into it what you are getting into. And also, you need to get a feel of it to really figure out if this field is for you or not. And how do you get a feel of that without, actually going to labs or actually going to hospitals and working? So I would say that start early if you are interested in medicine. I would say do volunteering work in hospitals, in research labs, do summer training, summer internships in the labs, so for example, write to scientists and, tell them, send them your CV and tell them that I am interested in, science because of this reason. And, I want to figure out basically whether I want to be a clinician or whether I want to be a research scientist. And to figure that out, I want to spend time in your hospital or research lab. So I think starting early and actually finding people who have done that, who have been through this path and talking with them, that's really important. And choose your college wisely after school, basically, because if you are interested in medicine, then you would like to get into a college where they have good research or hospital associated with them, and where they have a high rate of people getting into medicine from there, and where you could basically pick up the right courses. Those are needed to form the foundation for a career in STEM or medicine.
All right, thank you very much. So now we will be moving on to the audience questions. So I do not know what happened, but for some reason, we don't have a chat in Zoom. And I know some people are uncomfortable speaking out loud. So for that, we have made a quick Google form, which members can find in Facebook and I believe also Instagram. So you can go ahead and fill that out if you haven't already. And while you guys formulate your questions, there is one that I had while you're presenting. In CRISPR, when you cut and paste with the molecular scissors, can you take DNA from anyone and put it into anyone, or are there some restrictions on that?
You can put any DNA, actually. You can make a piece of DNA in the lab, an artificial piece of DNA, and even put that. You can take a gene out from something else and put it in there. The only limitation is the size, how big piece of DNA, but you can then, so right now the biggest size we can put in, so people were using viruses to put actually pieces of DNA into, because you need a method of delivery. So you have this CRISPR system, you have a piece of DNA that you want to put in somewhere, but how will you make it reach the right cell type? So people are using different ways of doing that. And I think currently what we can do is close to few thousand base pairs. So so DNA is double stranded, right? So and ATGC. So this is A is one base, T is second base, G is third base, and four. So if you have suppose ATGC continuously, that's 4 base of DNA. But if it's double stranded, so it's 4 base pairs. So similarly, we can change few thousand base pairs at once. But if it goes bigger than that, we can't do that right now.
So we have one submission, and the question is, have there been any major breakthroughs in your research or work in finding a cure for HIV?
Very nice question. And I just want to add before I answer this question that please feel free to ask as many questions as you would like. I love questions, and there are no silly questions, because sometimes the question, some of the silly questions makes us scientists think more deeply about that problem. And sometimes we get new ideas. So maybe your questions might give me more ideas about how to pursue some of my research questions. So breakthroughs. So the problem why we don't have a cure for HIV is what makes HIV virus so different is twofold. One is that HIV virus mutates very, very fast. It changes itself really, like coronavirus, the pace at which coronavirus mutates is nothing in front of HIV. Flu changes much faster than coronavirus. For flu, we have to take a vaccine every year. And because the flu virus changes so fast that next year that vaccine is no more effective, and you take a shot again. But for HIV changes so fast that you imagine. So suppose you take the viral diversity of the whole world for flu and compare it with one HIV infected person. In one HIV infected person, you'll have more mutants of HIV than in the whole world for flu. That's how fast HIV mutates. So the one problem is that it changes so fast. That's the problem with the vaccine design. The second problem is that once someone is infected, the virus goes inside the human cells, and it's an RNA virus. So it reverse transcribes itself into a DNA molecule, and the DNA gets integrated into our genome. So virus actually gets integrated into our DNA, and it can stay there without doing anything. So it can stay there silent, just sitting there quietly. And so what happens because of that is that, so when you are taking the medication for HIV, you kill all those cells which are actively producing the virus, because because the drugs can recognize these cells are producing virus and kill those cells, or our immune system can do that as well. But if there is a cell which is HIV infected, but it doesn't look any different from other cells, it's basically virus is not doing anything, just sitting there silently until a day, until when you stop taking the medication, and then those cells start producing the virus again. So that's why so I work with people who were infected in 1980s, and they still take medication for HIV every day. If they leave it for a week, their virus levels come back to the level 40 years, what were 40 years ago. So virus is there. So this is called latent virus or hidden virus. And people don't understand why the virus is hidden. Now, what I have done is I have developed a method using which, and people knew that virus is actually hijacking some of the host genes, some of human genes, to stay hidden. But which genes, people didn't know. So recently I developed this method using which we can actually delete all 20,000 genes in human genome, infect with HIV virus, and figure out which genes virus is hijacking. So we are slowly making a map of things, that these are the genes, this is how virus hijacks. And I think that is going to soon help us or other scientists in the field to actually find out new drugs which might bring this virus out of the hiding. And then we'll use the drugs that will kill that virus. And so we might be able to basically find a cure. So basically, I think my recent breakthrough is developing this technology using which we can actually delete all these genes and infect with HIV to figure out which genes it hijacks.
All right, thank you. Next question, what type of equipment is commonly used when analyzing DNA?
So a lot of different type of instruments are using for analyzing DNA depending on what you are doing with it. For example, one of the most cool instruments we use these days is, like, you might have seen some of those serials where people solve murder mysteries and all, right? So they say that, oh, this murderer was a brother of this person, or this is the father of this kid, or these sort of mysteries. And this show you all the structures of DNA, the helix type of structure of DNA. And this is not how DNA looks when you look at it in reality with your eyes, because you don't have that kind of resolution that you can see DNA bases and the helix. So we starting with some very basic instruments. Those are called electrophoresis instruments. So DNA is negatively charged, so we can actually have a gel-like structure, and we put some DNA in there, and we basically apply electricity. And because it's negatively charged, DNA, basically different size of DNA runs differently. So you can see, okay, this big DNA is here, small DNA is there, and other DNA. So that's a very basic technique. But one thing which we use very regularly is we use a machine called polymerase chain reaction, PCR machine, that can make thousands and thousands of copies of one molecule of DNA. So if you get one molecule of DNA from somewhere, you can make like million copies from that, and then do experiments with this million copies. Like, imagine you get a sample from a COVID patient, and you get some, like, that's an RNA virus. So you get a small piece of RNA from there. So you can use this instrument, the PCR instrument, to make DNA from that RNA, like just from one copy, and then make like millions of copy and do whatever experiments you want to do with it. Other thing we use very regularly is sequencers, the next generation sequencers, they're called. So because you can't see, when I say ATGC, GC, this DNA sequence, you can't see it. You can't see whether it's A after T or T after A. So, but with this sequencer, you can actually figure out what's the sequence of DNA. So you can read the code. Like, you can read in Microsoft Word, if it's written in Times New Roman, you can read it. If it's in different language, you'll not be able to read it. But so this machine, this sequencer, helps us read the code of DNA.
All right. So what will you do after you complete your postdoctoral training? Like, if I were to finish it, what would be my next step in my career?
Yeah. So I'm actually kind of transitioning from my postdoctoral training right now. I recently wrote a research grant, which was an independent grant, and I got it. So I'm slowly transitioning into an independent academic assistant professor title. And I'll be continuing to do research, but so there will be two things which I'm focused on right now. One is that starting my own lab, and basically hiring more people who work with me, so that building my own team. But also like some of the things which we'll find from these CRISPR experiments, the things you can cure. So basically getting patent for those molecules which we think you can target and cure HIV. And then if we find something which is really cool and which we think can cure HIV, or actually at least can give us that kind of treatment which you have to take once in few months, not every day, that itself will bring a huge change in the life of patients. So maybe starting while starting my lab, also starting a company, a biotech company that will focus on treatment of these viral infections.
Okay, thank you. Now focusing on CRISPR, how accurate and effective is the technology? And in your opinion, what are the biggest risks that come with it?
Yeah, it's a very, very revolutionary technology. So you think about it, that the kind of work I'm doing, we are not talking about even I'm not even talking about treating someone with CRISPR. I'm talking about using CRISPR to knock out genes and finding out which pathways, which genes virus hijacks, right? So we are going and asking very basic questions, and we have found out a tool using which we can figure out so many different phenomena, how something works, which genes are involved in a particular thing. For example, if you knock out a particular gene, then you see the cell becomes cancerous, then okay, that gene is a cancer controlling gene. You delete that gene, you introduce another gene, and what you do is increase the levels of proteins coded by that gene, and you see something else happens, and you figure out this is a function of this gene. And so in disease, in regular biology, you can figure out so much with CRISPR. But then again, the next thing is that in our lab, we are also using CRISPR gene therapy to treat patients, basically to find out treatment. So there's a family that have a particular gene mutation in their immune cells. So you have two types of immune cells, major two types of immune cells, B cells and T cells. B cells make antibodies, and T cells directly go and fight with pathogens. They kill them directly. So they're called killer T cells. So what we have found out in one of their T cells, in this family, in their T cells, they have a specific mutations that make them more prone to inflammation. So they have joint pain, inflammation, regular fever, and cold, and all those things. So we have figured out which mutation is that, and now what we are doing is we are taking out those cells from those patients, and outside in the lab, we are modifying those genes. And then we are trying to figure out whatever the defective function was, if that's getting restored. And if that gets restored, we're going to put that back into the patients so that they recover from this problem. Similarly, we are also working on treating cancer this way. So we can modify T cells to actually go and fight cancerous cells. So the T cells get trained, so they become like nano robots. So you train them to go and find cancer cells inside the body. So take T cells from the patient's body, and their natural function is to kill cells. They're killers, but they don't know, or they are not efficient enough to go and kill cancer cells. So, but you train them specifically, you change a gene in these T cells, and then gene directs them to specific cell types. For example, actually one of my nieces, she was 10-year-old and she was diagnosed with acute lymphoblastic leukemia. And she came to our hospital at UCSF here, and all type of radiation therapy and drugs failed. And we were worried that she's not going to make it. But at that time, what we did was like I'm using we very loosely here. Basically, doctors at UCSF, like clinicians, her doctors, what they did, they took T cells out from her body, and the cancer she had was a B cell cancer. Like her B cells, her antibody producing cells were dividing endlessly. So her whole body was full of these B cells, and because of which no other cell type was produced, and she was not able to breathe, and she was having all kind of side effects. So so those T cells were taken out from her body, and we changed a gene in those T cells in such a way that now those T cells started and finding B cells and killing them. So you put those T cells back, and they killed all the B cells from her body. And then she needed a bone marrow transplant to get B cells, because the T cells killed all the B cells in her body. And it's been 2 years, and she's in remission. She's doing well. She's now 12 years old. And so you can, with gene therapy, you can achieve such wonders. Now the problems, right? The problem, what you asked, the problem is that the CRISPR method can also have non-specific cutting. It's very rare, but like you want to cut DNA at a particular point, like point A, but it makes some very minor cuts at point B as well. And let's imagine that was a gene that was important for controlling tumor, and you cut that gene. So you might be treating a patient out of blindness, but you might give them cancer, and you don't want that, right? So you have to basically figure out ways to sequence DNA in such a way that you know that changes are made only at the place where you wanted to make, and not at any other place in the 3.2 billion base pair DNA sequence. So there are challenges with it, and that's why you don't want to use it for changing, germ cells. Germ cells are the cells which give rise to the next generation, right? So you don't want to change. So what we do is we take blood cells out from the body, T cells, and we modify them, put them back. But those cells are not going to go into next generation of to your next generation, right? But if you change cells that are going to go to next generation, you don't know what that's long-term effect is going to be, how that's going to be with the new in the offspring. New combinations of genes are going to come. And with this new combination of genes, what's going to happen with the change that you made? So we do not change germ cells. We only change cells that do not pass on to the next generation. And we are developing methods basically to figure out that there are no unintended changes in the DNA because of it. But that's always a concern.
All right, thank you. So another question is more opinion-based. So what do you think about anti-vaxxers, and how should we confront anti-vaxxer aggression?
Yeah, this is a thing which I'm dealing with a lot these days. I actually, during the time of COVID pandemic, when I saw a lot of people are not able to reach to the right information, I started making YouTube videos at that time explaining how vaccines work and all. And I got a lot of slack on internet. So if you go, very nasty comments on YouTube and all, because people they think that they say basically you are being paid by WHO and Bill Gates and all those people. That's why you're saying all these things. So sometimes people who are really, really against it, it's very difficult to change their mind, but there are a lot of people who are on the fence, and we should start talking with them first. And I think the way to deal with this is not to put everyone in the same category, thinking that these are just anti-vaxxers and they just don't want to believe on vaccines. You should ask them, what's the reason why you don't believe that vaccines work? What are the things? And then address their specific concerns. For example, the one person I talked with in my family, actually, and they were a little reluctant in getting vaccine early on. And they were very old, and I was concerned if they wait for 2 months and after 2 months realize that vaccines are worth it, by that time they might get COVID infection, they might not survive. So when I asked them what was their reason behind reluctance to vaccines, they told me that they don't have reluctance to vaccines in general, but they think they were developed too quickly. How can a vaccine be developed in 1 year? Because they have read that vaccines are developed usually in 10 years, 12 years. And so how come this happened in vaccine? So it means that they were not properly tested. And so then I told them, I explained, what is the vaccine development pipeline look like? So you do a research, you find out a new virus, you do research on for it many, many years, you understand how to make a vaccine against it, then you test those. And then once you have a vaccine, even to test that takes many, many years, because suppose what happened in 2003, the first SARS infection came and vaccine was made for it after a few years, but there were no SARS cases. How will you test a vaccine? How will you do a clinical trial? So the way you do clinical trial is that some people should be getting infection, right? And then you give a vaccine and you show that they don't get infection after getting the vaccine, they don't die after getting the vaccine. But if no one is getting an infection, you can't test that vaccine, you can't do clinical trials. Here we had so many people getting infected with this virus that we could do fast clinical trials. And then a coronavirus came in 2003, SARS-1. So we were studying this for last 17 years, 18, 16 years before this pandemic came, and we were lucky. If the SARS-1 would have been such more transmissible, we would have been a lot of problem. But we already knew that spike protein of the virus is the protein which we need to target for making vaccines. We understood, we had things designed for it, and then SARS-1 disappeared. But luckily that helped us prepare for this particular pandemic. And then new technologies, the mRNA technology is the latest technology, which is much faster to produce, much faster to scale up, much faster to change for new viruses. So that's how we could do it, all these things. So sometimes, it takes a lot of energy, a lot of patience, and it is time consuming, but I think it's worth it because all of us have reached to different demographics. Like I made those YouTube videos in Hindi because that's my mother tongue. And so I could reach to a very different audience. Whereas, you might be able to reach a different audience. So I think one logic which is very easy to give is let's look at data together and let's see the 5 billion people have been injected with COVID vaccines now in the world. And if the vaccines would have been causing any harm, in 5 billion, we would have seen a lot of trouble so far, and we haven't seen that, right? So vaccines are actually one of the most well-tested drugs we have right now.
Okay, this is probably going to be our last question. So I'm sorry to the people whose questions I couldn't get to. I will, if Dr. Rathore, you're okay with it, I can share your email so they can ask any questions.
Yeah, and I can, if people can I can stick around for some 10 more minutes and answer those questions if people want those to be answered now.
Yeah, out of respect for everyone's time, though, I'll have to make this the last question, sadly. Has an organism ever rejected the modified DNA, and if it has, what had happened?
This is a very interesting question. So if it's a foreign DNA that you are putting into an organism, it's possible. And because, so what happens is usually the antibodies are generated against, antibodies basically reject something foreign. They fight with it, they see it and they go. And that happens with proteins usually. And so what happens is in CRISPR-Cas9, I told you that this Cas9 protein actually is from bacteria, right? So it's not a human protein. So when you put it into humans, what can happen is the immune system can generate antibodies against this Cas protein and clear it off. So it might, so that's a way of rejection, right? So absolutely possible. People are trying to make these Cas9 proteins more human type, so changing their sequence so they become more human types and making them less immunogenic so they don't generate much antibodies, or figuring out way to deliver them directly into the tissue where you need it to be, so that our immune system doesn't see it before that, and by the time immune system sees it, it's already inside the cells, and by the time it's too late for immune system to reject it. So the rejection is a real issue. But there are ways to deal with it, and we are developing new ways every day.
All right, so that concludes this seminar with the guest speaker. Thank you so much to Dr. Ujjwal Rathore. Your work is very exciting, and I found this time to be very inspirational, insightful, and educational. So thank you for spending your time with us. We really appreciate it. Before everyone leaves, though, we would like to take a